Good morning, everyone. Thank you very much, Chair, for the really kind introduction. And first of all, thanks to the organizers for giving me the opportunity to introduce you to the GENIEUR network. And I have prepared kind of a wrap-up session. Where are we and where we want to go? For those of you who are not, these are my disclosures. I have basically none. So what is GENIEUR? GENIEUR is a network which started out as a COAST action, which ended last year, which stands for Genes in Irritable Bowel Syndrome Research Network Europe. It's chaired by me and my co-chair Magnus Simon from Gothenburg. And so far we have 27 countries from Europe involved, as well as 3 overseas countries. And basically, a lot of people in neurogastro, as you can imagine. You might see some familiar faces here, are involved in the management committee, and this has actually been taken, this photo, at our first meeting, at the FNM meeting in Bologna. So those of you who are interested to go deeper into GENIEUR and our output, you had this little booklet in your conference bag. If you're interested, just explore that a little bit, and there you also find a review which we have written among the members of the genetics working group. And what I'm telling you today is basically partly written also in that review. So irritable bowel syndrome, the pathogenesis is quite complex with multiple layers of complexity. And this is because extrinsic factors, so the environment like stress, trauma, sex, so the gender, smoking, diets, or lifestyle, as well as infections, shape, actually intrinsic factors, so our genetic background. In crosstalk with environmental factors, leads to the fact that also epigenetic effects take place, and also the gut microbiota in the crosstalk with the host genome contributes to different phenotypical features in the gut as well as in the brain, and via different axes which are corresponding, which leads to the fact that the gut and the brain are corresponding with each other, with the brain-gut axis as a major axis communicating between the both of them. And any kind of changes coming from the extrinsic and intrinsic factors lead to specific changes in that areas, shaping the clinical phenotype of the patient, IBS, as well as comorbid conditions like pain disorders, as well as psychiatric conditions. So far, quite some substantial genetic studies have been performed in IBS. 2 whole genome studies have been published. I won't go into detail today here, as well as several candidate studies tackling the serotonergic system, the immune system, the barrier function genes, neuronal function, very recently a gene involved in carbohydrate metabolism, as well as the bile acid pathway genes. And today, I won't go into detail in everything, but I will focus today onto the serotonergic system as well as on that part. So, why is serotonin important? As you all may know, more than 90% is synthesized by the enterochromaffin cells in the gut, and its release, as it has recently been shown, is stimulated not only by any kind of pressure, by, for example, a bolus passing by, but also by microbial metabolites. The serotonergic system is involved in many different physiological processes. It regulates GI motility, secretional processes. It's involved in the regulation of visceral perception, as well as it mediates pain, emesis, and anxiety control, among others. And actually, disturbances in IBS in the serotonergic system have been reported, and it's well known that serotonergic agents help some of the patients, but not all of them. And serotonin function is mediated via 7 serotonin receptor classes, HCR1 to HCR7, and all these facts led to the hypothesis in genetics that a disturbed serotonergic system may make people prone to impaired GI function and in turn then lead to the manifestation of the phenotype. The one polymorphism residing in the serotonin transporter gene, which we name in genetics SLC6A4, has been most intensively been analyzed not only in IBS but also in comorbid conditions. This is the so-called SL polymorphism residing in the promoter region, and the promoter region drives the expression of the gene, and if a certain polymorphism is present, this has functional consequences. I will come to that. And first associations of the short allele have been reported in psychiatric genetics to anxiety and depression, and later on in IBS, but not consistently in one subtype. Some found associations in diarrheic IBS, others in constipated patients, and other studies didn't find anything. So this is still controversially discussed. What we know from the psychiatric genetic studies is that the short allele seems to lead to less expression of the transporter, which in turn would mean that we would deal with higher 5-HT levels in the system. And studies from several groups worldwide, and among them the group of Micah Milleri, try to correlate the genotype of the patients with the levels of in this case, the serotonin transporter, and couldn't see any correlation. However, a more recent study from a group in India could nicely show that in IBSD cases, 5-HT levels seem to be increased compared to other subtypes, and this goes in line with the SS genotype, which very well fits with the hypothesis. Less serotonin transporter, more serotonin in the system. So, this polymorphism has been explored in pharmacogenetic approaches, and in the study from Micah Milleri, it was shown that the LL genotype goes along with pharmacoresponse to allosterone, and another study from China showed that the LL polymorphism is associated with improved symptoms, bowel movement, as well as the stool consistency. So, however, since this P1 promoter findings were quite contradictory, we thought about what else might cause these changes in 5-HT in the system, and we stumbled over a work of Gary Moore's group where they claimed that there exists another promoter driving the expression, which seems to act GI specific, and we then started out to explore that novel region for the first time in order to see whether we can correlate something here. And to make a long and complicated story short, we checked promoter 1 and promoter 2 driven isoforms and could confirm, yes, the P1 promoter driven isoform is not detectable in the GI tract. We ended up seeing that only the P2 promoter driven isoform is indeed expressed, and most robustly in the small intestine. We found a whole bunch of variants to be associated with female IBS, and in the meantime we ran a meta-analysis where quite some centers from the GENIEUR network were involved and could confirm an association of a SNP in this particular region where many other SNPs reside with female IBS. We further explored the functional relevance, and could see that it is indeed functional relevant and leads to decreased expression of the transporter. And we furthermore checked jejunal material of patients and could also confirm that expression levels in the jejunum are differently expressed in carriers of the variant compared to non-carriers. So this is currently wrapped up and hopefully published soon. My group has a long-standing interest in 5-HT3 receptors, and here we could show that 3 different polymorphisms could consistently be associated with diarrheic IBS, one residing in, again, a regulatory expression driving region in HDR3A. This is also functionally relevant and leads to an increased expression, and this one has also been in earlier studies associated with anxiety and depression and changes in amygdala activity. Another one, which resides in the coding region and is also functionally relevant, leads to a decrease in 5-HT3 receptor levels on the cell surface. Also with IBSD, and another one in the 3' untranslated region impairs microRNA regulation and seems to lead in a functional readout to increased expression of the gene. So all 3 of them are functionally relevant. And in the meantime, this one has been replicated in 2 studies from China, and the 3E one has been replicated in the very same studies from China. This locus has been, the HDR3 locus has also been found to be nominally associated in a GWAS from Mauro D'Amato's group, and we recently also replicated it in a study from the IBS Network Germany, and we have generated a whole bunch of additional genotype data which are currently in meta-analysis, and we are wrapping that up soon and hopefully also going to publish that soon. Furthermore, we checked out the 5-HT4 receptor gene and found another variant in the 3' untranslated region, also interestingly to be associated with IBSD. And this one also impairs microregulation, and in a functional readout we could also see if the variant is present. So look at this bar compared to the non-variant to the wild type, it also leads to an increased expression of the gene. So this is also functionally relevant, and then we furthermore thought it's worth to check out these microRNAs as well. Maybe they also contribute to the phenotype. And again, to make another long story short, in fact, we were indeed able to find microRNA 16 and microRNA 103, the ones which are most robustly expressed in the gut, to regulate the very same isoform, and that we came up with that particular model. So please have a look here. So in case we have a down-regulation of the respective microRNAs, this seems to lead to upregulation of the gene of the 5-HT4 receptor, and the same phenomenon takes place if a SNP is present, because then the 3' UTR escapes this microRNA regulation, which would then in turn lead to the same phenomenon. And this, of course, would have consequences in terms of increased mucus production, because more receptor is present, increased secretion and peristalsis, very well going along with symptoms which we then see in IBSD patients like accelerated transit, increased bowel movement, increased mucus release, and a very loose stool. And we were also very pleased to see that that microRNA levels also correlate with bowel movement and stool form. So if the microRNA levels go down, then the bowel movement increases, which also very well fits with our hypothesis. And this has been presented as a poster and is currently in revision, so also hopefully to be published soon. And now Another gene which has been explored by a group in the US, also in collaboration with partners from which are also involved in our GENIEUR network from Sweden, Italy, and Greece. And in this case, the SCN5A gene, which encodes a voltage-gated ion channel, was explored based on the fact that many patients with cardiac arrhythmias caused by SCN5A mutations also present with symptoms of IBS. And this, in this case, led to the hypothesis that disturbed neuronal signaling also may make people prone to impaired GI function. And what they did is they sequenced the gene. They found a whole bunch of mutations in 2.2% of patients. They found that they carry one of these missense mutations. And they also found that, and these are the ones which are highlighted in yellow and red, they found in functional readouts that the mutation leads to impaired function of the ion channel, and they detected, this was the case in 10 of the 13 detected mutations. These are the ones which are highlighted, and I'd like to draw your attention to this one, because this one, they found that it showed the strongest impairment in vitro. So this is a voltage clamp readout with no of the mutation in comparison to the wild type. And so if the mutation is present, we see different functional properties. And they also could show that mexiletine, which is a drug taken by patients with arrhythmia, is able to partially rescue the phenotype, and also that mutation carriers show, in fact, symptom improvement after taking the drug for a while, which is quite cool. So, and in a more recent study from Mauro D'Amato's group, his group actually checked out a gene relevant in carbohydrate metabolism, the sucrose isomaltase gene, SI, which is a desacharidase, and this was based on the fact that certain patients with sucrose intolerance suffering from congenital sucrose isomaltase deficiency show mutations in this particular gene, which lead to disaccharide malabsorption, which is characterized by diarrhea, abdominal pain, and bloating. And then a bell rang. Of course, these are symptoms which are very familiar in IBS patients. So another hypothesis arose that impaired function of that particular enzyme may also make people prone to suffer from disturbed GI function. And they checked out the particular gene and in CSID patients and found a whole bunch of mutations which, based on mutation prediction algorithms, seem to be functionally relevant. They furthermore checked these particular mutations in a genotyping assay in IBS patients, in particular the ones which are more common, and they could indeed find some of them to be associated with IBS, and this particular one they could robustly find to be associated with IBS. And so this confirmed that functionally relevant gene variants in this particular gene seem to increase IBS risk. And they furthermore followed that up in a functional assay and could prove if the variant is present, this leads to decreased surface expression levels. And they could also prove that it's functionally relevant in this case. and that it goes along with an increased symptom score. In another study, checking out in collaboration with a group in Barcelona, our group and the group of Javier Santos found that again in a RNA sequencing approach combined with a microRNA profiling approach, that two particular microRNAs are found to be downregulated in diarrheic IBS patients. And by doing in silico analysis, it turned out that both of them regulate genes relevant in barrier function, like claudin-2 and singulin. And we found that the particular microRNA, that they are upregulated, so in the opposite direction, as one would assume with an impaired microRNA regulation. If the microRNA levels go down, and then in turn the respective mRNA escapes regulation and goes in turn up. And in a functional readout, they could also confirm that the barrier function is impaired if they apply the respective microRNAs of interest. So less microRNA leads to increased decreased barrier function, and the opposite. So it leads to a leaky gut. And this also correlated with claudin-2 protein expression levels and singulin expression levels positively correlated with mast cell numbers, and in the opposite direction, this was the case with the respective microRNAs, coming up with the hypothesis that if the respective microRNA is downregulated, the target genes, which are tight junction proteins, are upregulated, and this leads to a leaky gut. So in summary, we can say that so far in the studies which have been published, so the robust studies, we found genes relevant in for psychological comorbidity, like the HCR3 gene, genes relevant for neuronal function, like the SLC-A4 gene and the SCN5A gene working in the brain-gut axis to be relevant, as well as genes working on spinal afferents, as well as genes and microRNAs relevant regulating epithelial barrier function to be involved. Furthermore, the sucrose isomaltase gene, which is relevant in terms of carbohydrate metabolism, leading to the fact that so far IBS-related pathways, based on the genetic and epigenetics findings we have, include potential pharmacogenetic and nutrigenetic targets, which is quite interesting. But, however, these findings have, of course, to be replicated in additional cohorts. The functional relevance in detail in vivo has to be explored. And, of course, you need appropriate model systems, indeed, to better understand what is going on here. And this brings me to the last slide of my presentation, what we envision how in the future genetics and epigenetics IBS research should be performed. You have seen in my very first slide that the pathogenesis is very complex. Multiple layers contribute, and it's not only the genes. I'm totally aware of that, and this became even more obvious in our network. That's why we came up with a detailed protocol, how we envision how to do a better job in the future, which means assess phenotypic features as detailed as possible with a standardized case report form for patients and control individuals, do deep phenotyping, and this is explained also in our booklet. I won't go into detail, and those of you who are interested, we have published a position paper stating that in a review article in Neurogastroenterology and Motility, where we in detail explain everything. All this patient-related data will go into a central database, And we also will sample certain biomaterials in order to enable us to generate genetic data as well as epigenetic data. And of course, we will also validate everything in tissue material. And all the findings will hopefully in the future, applying an integrative data mining approach, enable us to build up, based on the information gained, novel IBS subgroups, which we hopefully in the future will be able to offer a personalized treatment. So this is our vision. This is where we want to go. And with this, I'm at the end of my presentation, and I thank you very much.